Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, Health, Medicine, and Bioscience Edition. I have Hein Min Tun. He's a public health veterinarian. Received a PhD from University of Hong Kong. He went to Canada to pursue postdoc research at the Gut Microbiome Laboratory at University of Manitoba. He's a lab manager there as well. He worked there, looked at microbiome and resistome research in food animals, human, and the environment. Then he joined the SyMBIOTA research team at Department of Pediatrics and the University of Alberta to study infant gut microbiome and his health interest there. So we're going to talk about his current work. So Hein, thanks for coming. How are you doing?
I'm fine, yeah. Thanks for having me, yeah.
Oh, good. Where are you now, and what are you working on at this moment?
So I'm currently at the School of Public Health at the University of Hong Kong, and I'm also jointly affiliated to the Hong Kong U Institute Pasteur as a research assistant professor. I'm leading a research team here at the.
And what's the focus of your current research? Is it microbiome related, or is it different?
So yeah, I'm leading on the many microbiome research program and also antimicrobial resistant research. Yeah, both. So both
Okay.
both of like microbiome and also antimicrobial resistant research are quite interrelated each other too. Yeah.
Yeah. Well, how do microbes appear to become resistant to medicines? Is the mechanism, are there various mechanisms that are understood?
Sure, like, as like human being, like microbes also try to live in this world, so they try to change their genetic material if they from the like attack from the like antibiotics. So that's why they're trying to do, resistant to the antibiotics in different ways. So changing their genetic material, trying to escape from the different mechanism of the antibiotics when antibiotics are trying to attack the metabolism. So they try to resist them. Yeah.
Well, again, how does a microbe become resistant to a drug? What are some of the mechanisms that have been observed?
So, antibiotic resistant is this like, especially the kind of evolution of the bacteria. So they have a kind of certain type of the mechanisms that is trying to change their genetic like structure, trying to escape from the recognition of the different antibiotics. For example, like some antibiotics are targeted to the different structure of the bacteria, but they try to change their genetic material to change the recognition from the different antibiotics. Yeah.
Okay, so what are you trying to figure out? How to stop them from becoming resistant, or a drug or an interaction that will overcome any resistance? Like what's the thought process there?
So now my research area is not trying to very basic science. I'm trying to answer the questions how the microbial community changes is related to resistant, antimicrobial resistant, especially the gut microbiome. So how the changes in the gut microbiome, how the dysbiosis in the gut microbiome is causing our colonization resistance, changing our colonization resistance against two different antibiotic resistant bacteria. So we have a lot of resistant bacteria, in our gut, but our level of the resistant, bacteria is different among the different individual. So, and we also hypothesize that probably that is related to our gut microbiome, the community structure in our guts. So that's why we want to try to understand how the gut microbiome interrelated with the resistome level in our gut.
Okay, again, are there any specific mechanisms that you've observed by how resistance arises, or what are some of the dynamics of it? In order to figure it out, I think we'd have to understand.
Sure. Sure. In our kind of preliminary finding, we found that reducing the diversity in our gut microbiome is one of the factors that is causing the colonization by the different type of the resistant bacteria, such as like ESBL bacteria, extended spectrum beta-lactamase producing bacteria. So that's why we think like the disruption of our gut microbiome, changing our gut ecosystem, and also that is impacting our host defense, host immunity, that is causing our colonization, resistant against the different resistant bacteria. So one of our interesting finding is this in reducing the diversity, especially among the butyrate producer in our gut, that is causing colonization, reducing colonization resistance against the ESBL bacteria. Our hypothesis is really reducing the level of the butyrate, where we also causing the reducing the level of the butyrate in our gut, and that is causing our the level of the butyrate level in our fecal, in our gut, that is causing our epithelium system change in the area. So that is causing a colonization resistance. Actually, this is very early kind of concept that we need to do more mechanistic study in this finding.
When you say diversity is reduced, is that diversity of function to create a certain metabolite, or is it just diversity of strains? And if so, is it strains that are close, or are they more taxonomically different?
So the first information we found is the overall diversity reduced. So the overall diversity reduced is not always good for the microbiome. In most of the study, you can see there's microbiome research has been reported that the reducing the diversity is correlated to certain different type of disease. But also for the antimicrobial resistance, also we found quite similar phenomena. Reducing the diversity, overall diversity is a problem. But when we are looking at very detailed level, we found that the reducing level of the butyrate producer in the gut is correlated to the colonization of the resistant bacteria. So we don't understand how the butyrate is we know that butyrate is, providing the energy for our colonal site, this epithelial cell in our colon. But we don't know why that is, causing the colonization resistant. Changes in the colonization resistant. We need to do more research on that.
All right. So, in particular, into the dynamics of the microbiome itself, at least the gut microbiome, have you figured out any particulars? Any functions that are critical to human or mouse life, or what has been figured out instead of this general? Okay, well, there's a microbiome and we're studying. What are some of the particulars you're looking at figuring out?
So another big project that I'm working on, it is we try to understand what is healthy microbiome. So when you are going to ask this question, this is kind of $1 million question. If you go and ask every microbiome researcher, definitely they cannot answer on that. So healthy microbiome I think everybody aim to understand what is healthy microbiome is. So this is also one of the objectives for my research group too. We try to understand what is healthy microbiome, but we are using the kind of different approach from other groups. So we're trying to look on the very early life microbiome development. That is trying to understand how we we inherited our microbiome from the mother, and also how that developed in the first 3 years of life, and that is also reflect to our healthy gut microbiome. So we are trying to understand so many things, how different type of factor is influencing on our microbiome development in early life, and how that is also contributing to our healthy gut microbiome. So that is one of the area that we are working on.
Well, if there's a lot of factors, which factors are you starting out with to elucidate? There's so many.
Of course. So I work with the Canadian health bird cohort study, and we try to look at in different factors, like, for example, like how the maternal factor like maternal obesity, maternal diabetes, that is impact on the baby microbiome development. And also we are looking at the different environmental factor like pet exposure, how that impact on our gut microbiota development in the early life, and also exposures to different disinfectants or like different chemical exposure, how that is impact on our gut microbiota. So we look at like several exposome, that is impact on our gut microbiota development in the early life, especially the first 3 years of life. Yeah.
So what, are you using a mouse model, or what kind of model are you using?
We are looking at the human sample, especially the population-based sample. So we know that like the mouse model is not the best model for gut microbiome study. Of course, we are limited to use for that. But my research is mostly look at on the population data. So we work with like different bird cohort. So one is the Canadian bird cohort, and also we are now working with the Chinese bird cohort study, try to understand how the microbiome development in the population data. And of course, in population data, not always clean as like mouse data, because mouse is you are doing in the lab, and then you have a lot of confined environments. But population data, you cannot do that. So, but of course, we are using different like statistical approach and also machine learning approach, trying to understand how the dynamic of the microbiome development in the early life in different population setting. And then that way we try to understand how the healthy microbiome should be look like.
So what have you noticed in the microbiome of, you're starting at 1 year old or earlier? Like, what does it look like in early lifetime?
We recruited the mother at that trimester of the pregnancy, and we try to follow that until the 3 years of the baby. Yeah. So it is kind of longitudinal cohort study. Yeah.
How often is the sampling, and is it just,
Yeah, sure. So we try to, like, in different cohorts, we have a kind of a little bit of different sampling strategy. So for the Canadian cohort, we try to look at the fecal sample at the 3 month old and also 1 year old. But for the Chinese cohort, we could, because we set up the cohort later on, so that's why we have a kind of chance to collect more samples. So we collect the meconium sample. We also have a kind of a week-old sample, a month, and also 3 months, 6 months, and 1 year, and also 3-year-old sample. So we have kind of more sampling time point for the Chinese bird cohort. So we try to look at in different window time in order to look at for different correlated with the different factor. So how different exposures impact in which time point. So that is to try to understand the development of the microbiome, as well as we also look at the development of our immune system too. We measure the certain immune marker like secretory IgA in the fecal sample, and also some metabolite sample. So we try to understand a more system approach to look at the microbiome development in the early life. That way we could try to understand the question of what is healthy microbiome look like.
Okay, so what have you seen in these longitudinal studies? Less diversity, a completely different set of microbes? Like what have you seen?
Of course, we found very interesting things. We try to look at the trajectory, development, microbiome development trajectory in early life. There is this different type of the microbiome developmental trajectory among our study infants. Let's say we could find 4 different type of trajectory, but among 4 different trajectory, we only found one trajectory is kind of related to the some of the outcome like the allergic disease like food sensitization, but not other trajectory. So it means that even you may have a different trajectory, but doesn't mean that your microbiome is not, not round, not really in the round way of the development. So, but that we try to understand that, how to interpret on the developmental trajectory of the microbiome in connection with the different disease outcome. So probably some microbiome development trajectory may be associated to some certain disease, but not for other disease. So that is also very clear that, we know that certain microbial taxa, that is a kind of risk for the certain type of disease like allergic disease and asthma, but not for the obesity. But obesity is a different way of the microbiome influencing factor. So we try to look at the different trajectory associated to different outcome.
Okay, is there any generalities you could see in babies and young humans that what does the trajectory look like? What does that mean? There's just less diversity?
So generally, we have a very, low diversity in our early life compared to adult microbiome, of course. But baby were born mostly by the, most of the bacteria in our early life is the proteobacteria. So that proteobacteria is like decreasing, the abundance is decreasing, from time to time. But at the same time, the bacteroidetes and firmicutes should be taking up our microbiota, and look like a kind of adult-like microbiome. And we also found that bacteroidetes play a huge role in the developmental trajectory. We look at that some of the depletion of the bacteroidetes in early life is associated with many disease outcome like food sensitization and also delayed neurodevelopment. So that's why we think the bacteroidetes play a significant role in our early life. But we can also look at why we have a lower bacteroidetes in the early life, because those kids who are born by the C-section, they have a lower bacteroidetes. Those kids who are fed by the formula, they are also lower in bacteroidetes. And that's why in epidemiological study, we also found that like C-section formula feeding, that is also associated to the same disease outcome. So that now microbiome is explaining why the epidemiological evidence are valid, because microbiome play as a kind of mediation role in this kind of association. So we thought that having kind of bacteroidetes is in the early life is much better than without having the bacteroidetes. So it's really important. That will also be a kind of room for the intervention study in the future. If we give more bacteroidetes, for the baby, we can be used as a kind of probiotic in future.
All right. Did you look at only C-section babies, or did you look at vaginal versus C-section, and then breastfed versus not breastfed? What were the main divisions?
So we look at in the population data, this is kind of general population data. We are not discriminated to only C-section and the vaginal born babies. So we recruit as all baby. So of course in our data, is included both vaginally delivered baby, C-section delivered baby, as well as the breastfed baby and also formula fed baby. So we try to answer this complex question in the population data, not like the mice model. We just only test on two factor or one factor, but we are trying to understand the multiple factor, how they are connected each other. So of course in our cohort, we have a different type of baby, who are breastfed or formula fed or C-section baby or the vaginally born.
Okay, so did you see a very big difference between vaginal versus
of course,
cesarean? and
So we that's what I mentioned now, like because like we found that there is a significant differences between the vaginal born baby and also the C-section baby. Of course, C-section baby have like totally almost quite, different from the vaginal born baby, and their developmental trajectories are different. But however, like maybe breastfeeding can compensate that, kind of differences, but it cannot be completely compensated, that based on our finding. If you are born by the C-section, of course, you will have a different microbiome, at the beginning. But breastfeeding can compensate in some level, but not totally. So that's why we are trying to look at what other factor can still compensate for the C-section baby.
Okay. Are you seeing a dramatic difference, or is it more of a slight difference in the two?
So the differences for the C-section is early life is much more, earlier time is much more significant, but compared to the later time point. But of course, later time point probably they can be compensated by the other nutritional factor like the different solid food introductions and also other environmental exposure that probably that still have some compensatory effect. That we could see more in the early life. That we also found that differences in early life is associated to the disease outcome.
Oh, what kind of diseases seem to affect cesarean section babies and non-breastfed?
Okay, so we are looking at in the allergic disease and asthma, so mostly on that. We also find the C-section baby have more likely to become overweight, and that is a very interesting story we found. So even though like if the mother is an obese mother, their risk is even double to become overweight.
Okay. So what's is there any way to, once a baby is born by C-section, is there any way to modulate its microbiome to go back to a healthier state, or not really?
No, actually, there is like, so that is what I mentioned before. So if the baby are born by the C-section, like if we cannot avoid it, so probably we should try to breastfeed to those baby, at least like exclusively breastfeed for 6 months. And we also try to make those baby to expose to more, nature environments. And, that is, we also found in our study that if baby are living in the more nature or related environment or green space environment, they are more likely to have exposed to more microbe. And that is also a kind of, compensatory factor for their microbiome development. So, and also, based on my research published in 2017, those like C-section born babies should have a pet for sure. So to make sure that they can expose to more microbe, and train their immune system in a more like balanced way, that they will be much more similar to the vaginally born.
Okay. But again, if a baby's born C-section, you can, breastfeeding will help. Is there anything else that can be done? I've heard about vaginal swabs, maybe a way to, to make sure that there's
so
right microbiome.
yeah. So there is this research by the Maria Dominguez Bello from the New York University. She's trying to answer that question. So, and some of the study, they also find that like vaginal seeding is the one of the way to compensate the missing microbes in those C-section baby. But we have to be very cautious when we are doing the vaginal seeding. Vaginal swabbing for those C-section baby, because they can also transfer some undesirable microbe, or pathogenic microbe from those vaginal swab to the baby. So we have to be very cautious about that. And how standardization of those methods also should be also pay attention on that too. There is this not much standardization for that kind of method too. So we need more research on that area in order to apply that in the real situations. Yeah.
Okay. So, Hein, what do you think is going to be possible over the next few years? Any breakthroughs coming?
So for the my research group, we are trying to understand the what is the healthy microbiome, but using this kind of early life. So we probably in the next few years we trying to we were more understanding on the what healthy microbiome look like. Not only by looking at our different bird cohort, we also trying to do the meta-analysis from different bird cohort study across the world. So we're trying to collaborate with different other collaborator from the European partner and also other North American partner to try to find any consistent finding across the different populations. So that we definitely will understand more on the healthy microbiome, how healthy might look like. And another area we definitely could contribute to the field is like the neurodevelopment. So we're trying to understand how the gut microbiota, impact on the neurodevelopment of the baby and also the mental health. So probably we could also see some exciting new how our gut microbiota can also impact on our mental health and behavior and also neurodevelopment. Then that would also be interesting.
Okay, well, very good. Hein, what's the best way for listeners to find out more about your work?
So probably definitely try to follow at my publications and also try to follow in my website, and probably we will try to disseminate all our research finding in our website, so probably they could follow out on that.
Okay, well, very good. Hein, thank you for coming on the podcast. I appreciate it.
Thank you. Thank you for the time.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And be smarter than everybody else? Become a premium member at findinggeniuspodcast.com.